Hello everyone, welcome back to another episode of Tony's Insights. This video will be on nucleic acid metabolism. Alright, let's dive right in. Okay, so I said that we're looking at the synthesis, how AMP and GMP is synthesized from IMP. So let's say, let me go a little heading. Synthesis of um of AMP and GMP from IMP. A little recap. I said that IMP is the first nucleotide that is formed during the novo synthesis. So from IMP, IMP will now represent a branch point where AMP is formed and GMP is formed. But now let me tell you the interesting thing about this pathway. The interesting thing about this branch point is that for AMP to be formed by AMP is adenosine monophosphate, GMP is guanosine monophosphate. They are nucleotides, right? There's a phosphate group, there is the sugar in adenosine, and there's a pentose sugar in adenosine, and then there's nitrogenous base, which is adenine. So the nitrogenous base of AMP is adenine. The nitrogenous base of GMP is adenine. The nitrogenous base of guanosine is guanine. So when this guanine acts up with a pentose sugar, the pentose sugar, and still acts up with the one molecule of phosphate group. That's when GMP is formed. The same thing as adenine with the pentose sugar, and then with that's pentose sugar, that's the ribose. That's ribose, yes. And then with one molecule of phosphate, that's to form AMP. So you understand. Same thing as IMP. IMP is from the nitrogenous base of IMP is hypoxanthine. So this actually is hypoxanthine. You understand? This is hypoxanthine, the nitrogenous base of IMP. So now, okay, so let's look at the structure of IMP. This is the structure of IMP. Remember, this is the structure of IMP. Sorry. Remember that this is a purine ring. So there's a pyrimidine part of it, and then the imidazole ring that is fused. Sorry, the imidazole ring that is fused. There's a nitrogen here, and then like this. There's a nitrogen here, there's a nitrogen here, nitrogen here. So inosine monophosphate has at this carbon, there's a carbon here, right? All these please represents a carbon. So here is a carbon, here's a carbon, here's a carbon, here's a carbon here, here's a carbon here. So all of them represent a carbon. So at this carbon here, there is so I just want you to understand this bit. There's a carbon, just like I explained to you in the previous video about how this type of structures work. So there's a carbon here. You understand? Okay. So now for inosine monophosphate, remember there's a ribose 5. This is ribose 5 phosphate. Let me just write it like this: ribose 5 phosphate. So the ribose is the pentose sugar, the phosphate group, and then the inosine, the hypoxanthine. This is hypoxanthine now. They now combine, this all combined is IMP, inosine monophosphate. So for inosine monophosphate, at this carbon here, there is an oxygen atom that is doubly bonded. So let me explain to you how I normally draw these structures. Is that first of all, you put a double bond here. You remember how that carbon has to bond with 4 atoms. There has to be 4 bonds with carbon. Nitrogen, there has to be 3 bonds. So the carbon here, there's a carbon here, right? It's bonded to 1, 2, 3, 4. So it's complete. The carbon here is bonded to 1, 2, 3, and 4 as well. So it's complete. The nitrogen here is bonded to 1, 2. So there's actually a hydrogen here. But you don't have to show. You can either put the hydrogen here, but the hydrogen is implied. So you don't have to like kind of show it. Then the nitrogen, okay. So now this I normally use is that if there's a double bond here, there won't be double bond. Double bonds don't come close to each other. If there's a double bond here, the next double bond now will leave here and come here. So nitrogen is actually bonded to 1, 2, and 3. So actually there's no hydrogen here. Sorry, there's no hydrogen here. So there's a double bond. The nitrogen here is bonded to 1, 2. You understand? 1, 2. So then there's actually a hydrogen here. Yes. So there's a double bond here, meaning that there won't be a double bond here, there won't be a double bond here. The next place there will be a double bond is here. Here. Yes. It will be here. So this is the structure of inosine monophosphate. The carbon that is here is doubly bonded to oxygen, and this is the bond. This is so 1, 2, 3, 4. So this is the structure of inosine monophosphate. So now let's now look at, in my video on nucleotide, I'll really explain structurally how nitrogenous bases, how different structures of nitrogenous bases, and how to best learn the structures. So now let's look at the branch point. The first thing that is formed in the formation of AMP, the first thing that is in the formation of AMP, this is IMP, remember, this is IMP. So the first thing that is formed is AMP. So if you want to look at how AMP, for the sake of the board, let me just, if you want to look at AMP, so AMP is just that you form, it should be as a nitrogen here, a nitrogen here. So actually, aspartate will come in here. There will be an aspartate. This is ribose 5-phosphate. Ribose 5-phosphate instant. So aspartate will come in here. Aspartate will come in here. Aspartate is an amino acid. It is coming in to donate its amino group. So aspartate will come in here. So this right here is aspartate. Aspartate will come in here. Aspartate will come in here. Remember that I told you that the interesting thing about synthesis of AMP from IMP is that IMP to AMP requires GTP, but IMP to GMP requires ATP. So, like, it's a way of regulating the amount of ATP in the body, amount of GTP in the body. When there's excess ATP in the body, that means IMP will favor the production. That means IMP will favor the production of GMP, where there's much ATP in the body. But when there's a lot of GTP in the body, it will favor the formation of AMP. Understand? So, no, sorry, when there's GTP in the body, favor the formation of this GMP. So say this AMP, when there's a excess of GTP. So because right now we need plus GTP. Understand? So GTP is required, aspartate is required for this reaction to now occur. So what comes out is GDP plus, that's guanosine diphosphate. This is guanosine triphosphate plus the inorganic phosphate. So all this came out. So the name of the enzyme that will catalyze this, because aspartate will come in. See how aspartate comes in. This is okay. This is go. Okay. This is just order. This is so. This is how aspartate. So the one on green is aspartate. So aspartate will come. You know the point of aspartate. Aspartate coming in is just to donate this amino group here. So the next reaction, this reaction here is the formation of adenyl succinate, right? Adenyl succinate. So that's adenyl succinate. So the name of this enzyme now will be adenyl succinate synthetase. It requires energy because it requires energy, so it's called synthetase because it was formed. The name of this compound is adenyl succinate. Yes. So now, when there's now a break at this point now, it will form AMP. Understand? It will form AMP. That's all. So a break at this point will form this. There's a double bond here. Remember, there's a double bond here. Double bond here. Double bond here. Double bond here. Remember, yes. So this here now is AMP, adenosine monophosphate. This is AMP, adenosine monophosphate. Yes. Okay. Remember there's a ribose 5 phosphate here. Remember, there's a ribose. There's a ribose 5 phosphate here. So this whole thing is what? AMP. This whole thing is AMP. And what will catalyze the break at this point is adenyl succinate lyase. Okay, yeah. So the name of the enzyme catalyzing this step is adenyl succinate lyase. Because there was now a breakdown. So you might be like, where did this one now go? This one here, this right here. Where did this part right here, where did it now go? It left as fumarate. That's correct. It left as what? Fumarate. It left as fumarate. So this represents how AMP is synthesized. Let me just go through it again. This is structure of AMP. The characteristic is that at this carbon here, there's an oxygen doubly bonded to it. So this is IMP. So when first thing that will happen is that aspartate will come in. Aspartate is coming. It will aspartate and this IMP will together form adenyl succinate. Understand? Adenyl succinate is aspartate plus IMP. So the point of that aspartate is coming in so that it donates amino group. So see the amino group here. See the amino group here. So see the amino group that is donated. So when this one now will now leave as fumarate, leaving behind only AMP. So this right here is AMP. This is synthesis of IMP from AMP. Require GTP, energy in the form of GTP. So now let's look at this side. This is how GMP is formed. Let's look at how GMP is formed. Okay, so GMP is formed by the first thing that will happen in GMP is catalyzed by IMP dehydrogenase. This step of reaction is catalyzed by IMP dehydrogenase, inosine monophosphate dehydrogenase. Let me write that. Because so as I've said that it's inosine monophosphate dehydrogenase, you should know that NAD will be involved. Dehydrogenase, NAD, nicotinamide adenine dinucleotide will come to remove the hydrogen. It's the coenzyme that will come to remove that hydrogen. So NAD, you will see it, NAD will come in and NADH plus a hydrogen atom will leave. Yes, because the hydrogen atom will leave. So I'm telling that this reaction is catalyzed by IMP dehydrogenase. We'll soon see what is formed. Dehydrogenase. Yes. IMP dehydrogenase. And also, actually, H2O also entered this reaction as well. H2O entered to donate its oxygen atom. You see where the oxygen atom enters in this reaction now. Okay, so I'm redrawing this IMP, but I'll show you where the oxygen atom comes in. But this, what is formed now is, the name of the enzyme is what? Xanthosane monophosphate. That's what is formed now. Xanthosane monophosphate. So actually, the oxygen atom will come to this carbon here. There's a carbon here, right? The oxygen atom will enter here. So it will doubly bond to here. But do you remember that rule I gave you that two double bonds should not be together? So that means this will then clean, there won't be double bond here again. There won't be double bond here again. Meaning that here now has a hydrogen. Because nitrogen has to be bonded to 1, 2, and then 3. Here it was bonded to 1, 2, and 3. Take time to assimilate all I've been saying so far about the structure. So now it is bonded to 1, 2, and 3 as well. So with the oxygen here. Do you get this? Do you get this? Okay. So I'm saying that the name of this structure is xanthosine monophosphate. This is xanthosine monophosphate. And as I've told you, it is catalyzed by IMP dehydrogenase. Inosine monophosphate dehydrogenase, because NAD came in and took away a hydrogen atom. You understand? So the next step of the reaction is an amino group will then come in. Yes, an amino group will come in to form this GMP that we're talking about. So an amino group will come in to form GMP. So let's withdraw this. Okay, this is ribose 5-phosphate. So an amino group will then come in here, right? An amino group will come in here. So this amino group is coming from glutamine. The amino group is coming from an amino acid called glutamine. Understand? So this oxygen is still there. Remember, this oxygen is still there, and there's still a double bond here. There won't be double, as there's no double bond here. This one will kind of return, and then remember there's a nitrogen here. There's a nitrogen here. That's correct. Remember there's a double bond here. Yes, because two double bonds can't come together. Just remember. So this right here is now GMP. GMP. So GMP requires ATP. Remember when I told you that GMP, so when there's excess ATP in the body, GMP is favored. GMP is favored. Synthesis of GMP is favored whenever there's ATP. So look at this now. So let's look at what came in and what left. I told that glutamine will come in to donate its amino group. And when glutamine comes in, what glutamate is glutamine that does not have the extra amino group. So glutamine comes in, glutamate then leave. If you understand this type of reaction that went on, if you look at my video on synthesis of non-essential amino acids, look at that video. So you understand what happened there. So then for this, okay, yeah, ATP will come in as well. ATP will come in, and then adenosine monophosphate will come out. So will it be adenosine monophosphate will come out as well as 2-phosphate group, PP, pyrophosphate. So I was saying, what do you think the name of this enzyme here? What do you think the name of the enzyme that forms GMP, that synthesizes GMP? It will be called GMP synthetase. Very simple, GMP synthetase. It will form GMP synthetase. Okay, remember this one was ordinary succinate slyase. Yeah, GMP synthetase. Yes. Okay, so yeah, so that is it. That's all for this video. So you understand everything that went on. The first one, catalyzed by IMP dehydrogenase, there was removal of the hydrogen, these two hydrogens, because H2O came in now, the two hydrogen where it left actually, this oxygen will then come and bind here, and then this glutamine will come in to replace this part here. To replace this part here, add this amino group here, and then form this GMP synthesis. Then both this one and this step, they require energy. This one requires energy in the form of ATP. This one requires in the form of GTP. So this is the end of the first part of this video on synthesis of nucleic acid metabolism. Thank you. So please comment on the next video you'd like me to do on, the next topic you'd like me to do on. Thank you. Don't forget to like, comment, share, and God bless you.